Anything interesting about microscopy and light so far?
Linear systems.
This is very mathematical so far? Okay, so I'm going to talk about light sources, basically. So there's lots of light sources. Anybody know a light source that's available in a microscope or just in general everyday life? What's that?
Mercury lamp, LEDs.
Yeah, very good. So basically anywhere you see light coming to you from an artificial source that's also used for microscopy generally. So anybody heard of fluorescence microscopy? Have you all studied any fluorescence microscopy before? Okay, so I'm going to be talking about fluorescence microscopy. And that's a special type of light microscopy that's really, really, really widely used. And it gives you the special property that's really, really great of molecular specificity. So you can look at specific molecules. That's what's really great about it. It's compared to, if you think about shining light through an object, which is like transmission microscopy. This gives you something that's beyond that. It's beyond a structure. It tells you specifically a molecule's there because a molecule has to be fluorescent in order to detect it. So does anybody know what fluorescence is? What is fluorescence? Can anybody define it?
Observe certain wavelength light and emit ultraviolet.
Yep, exactly. It's some kind of emission of light resulting from a higher energy state of a molecule. And it all derives from a special molecule. You have to have a special molecule that will give off light to do this. And this is what we make. This is what we do in our lab, is we make fluorescent molecules. And so we could tag them to biological molecules, and so we could see biological molecules that were intrinsically invisible before. So we could study basic biology at the molecular level. So that's the key thing that fluorescence microscopy gives you the capability of doing. You can study specific molecules microscopically. So we work with, so obviously there's tons and tons of types of fluorescent molecules. They're all around you. So the molecule fluorescein, which is what people inject into your eye in order to see things in your retina when you go to the optometrist, and molecules like green fluorescent protein, which you can express in cells. They all give off light. And so the ones we work with are nanoparticles. And so nanoparticles have a lot of special attributes. And for fluorescence microscopy, you get a lot of great things that come out of them. So I'm going to take you all through nanotechnology first. And this could be really open-ended. So if anybody wants to talk about any one particular aspect of this, just stop me or raise your hand and say that's interesting or it's not. So this is going to basically talk about quantum dots. So quantum dots are kind of the focus of our research, and they're great fluorescent nanoparticles. So nanotechnology is this great field that's been around since it's been really defined as its own independent field for in the range of about 20, 30 years now. And you think about materials having a specific type of property, like a semiconductor allows you to do a whole bunch of things in electronics, and metals allowed you to have conduction. When you think about things on the nanoscopic world, everything depends on size. And so you have properties like a metal, which can change. It can become a semiconductor, it can become an insulator. So the scale of nanotechnology is around this. If you think about a molecule that you study in chemistry, which is in the range of about angstroms to 1 nanometer, it's much larger than that, but it's also much smaller than what you have in biology as the basic functional unit of biology. Most of that is in the range of hundreds of nanometers, like a virus or a bacteria, and a cell. Cells are usually around several microns to tens of microns. Between those two ranges, the chemical world and the macroscopic world is the nanotechnology world. So that's in the range of about 1 to 100 nanometers or so, roughly in that range. And in that range, it also is the same size range as proteins. And so proteins are like the functional unit of biology. They do all the functional useful work in a cell and in tissue. And so we're basically trying to develop inorganic materials that kind of mimic this and make use of all the properties of proteins that are so great in biology. And so here are like the classical ones that you'll hear about. So liposomes, dendrimers, gold nanoparticles, gold nanoshells, quantum dots, fullerenes. These are all somewhere in the range of 1 to 100 nanometers. We're going to mostly focus on quantum dots. So what do you get when you make a gold material, which is normally, like if you think about a film of gold or a block of gold, if you shrink it down to the size of a nanometer to 100 nanometers, its properties change. And that similarly happens to almost every type of solid state material. If you keep shrinking and shrinking and shrinking it, cutting it down into smaller and smaller chunks, at some point those properties that it had originally have changed. And so when you think about what would happen to all these different materials, like magnetic materials, metallic ones, semiconductors, catalytic, organic materials, they all get this new emergent property that arises when they get onto this nanoscopic size range. So magnetic materials become what's called superparamagnetic. That means they only have like one magnetic moment per entire structure. These magnetic materials are like iron oxide, and these are actually used in clinical medicine now. You inject tiny particles of magnets inside the body, and they can be detected by MRI. And so they're used as MRI agents to see where things are in the body, especially for looking at where blood goes in the body. Metals become something called plasmonic. Has anybody heard of plasmonics before? So it's really interesting stuff that goes on in plasmonics. It's a very, very diverse field. One of the key things about it for a nanoparticle that you might put in biology is that effectively all the electrons are on the surface of the material, and they can collectively resonate with light. And because of that, if something changes on the surface, then that will change the resonance frequency. And so you can detect binding events just by having something bind or desorb from the surface. So that's really useful. Semiconductors, which I'll talk about mostly today, have experienced this type of a phenomenon called quantum confinement. And these are like quantum dots. So quantum dots are a type of semiconductor, and they experience quantum confinement. That basically means you can change all their optical properties by their size. And so forth. You can look through these slides. I'll send these to Gabby so you can have copies of these as well. So basically what you get is these emergent properties that were not there originally for the bulk material, but they have these new properties that arise when they become in the size range of like 1 to 100 nanometers. Yes?
Are you going to expand on that quantum confinement?
Yep. Yeah, I'll do that right now. Well, just really briefly, nanotechnology basically, it's a field that actually is taken off into an industry now, and so a lot of this derives from cancer medicines. And so the cancer medicine, nanomedicine market is expected to be roughly in the range of $100 billion by the end of this year. And that's basically nanoparticles that aren't made out of metals or semiconductors, made out of drugs basically, and they could deliver things to the body. And so quantum dots, which we'll talk about, also have a lot of great industrial properties already as well, and they're used and sold commercially. Okay, so quantum confinement, this big property that you get out of quantum dots. First of all, let's talk about the structure of what these look like. So a quantum dot is a semiconductor, like I said. The great property that comes out of it is fluorescence emission, and so you can see them really easily. So this is what they look like. This is a schematic drawing of a quantum dot. So what you can see here is that the core of this material is a crystal. It's got a periodic array of atoms in a crystal. And it's got this coating on it. And this coating is supposed to depict organic molecules. This is supposed to be something like an alkane, which is just a hydrocarbon. And one end of it binds to the surface. These are the ones that we use. This is a variant of a quantum that's used in biology for biological imaging. It's called a colloid, which means that it's a suspended piece of solid in a liquid that we could put into our bodies, or we could put into cells, things like this. There's another version of this called an epitaxial quantum dot. And that one is derived basically from the microfabrication industry. This is what's deposited on the surfaces of like silicon So you can make LEDs and devices. And so these are two of the same types of material. They're both made out of the same thing, but they can be made as like a solid material grown on the surface, or it can be made as a dispersed fluid phase. And so clearly, if you want to interrogate things in cells, this is the only one to use. However, these are in use. These are in use in devices. This is an electron micrograph of a single quantum dot. And so this is a scale bar that's 5 nanometers. So you can see this whole thing is about 10 nanometers. And so in electron microscopy, you can only really see the optically or the electronically dense materials. And so this is just what you see here is the core. You don't see this shell region because it doesn't really have a lot of electrons compared to the core region. So this is kind of a depiction of what the whole structure looks like that we make today for biological use. It's got a core with multiple types of shells on it. And so this is a multi-step engineering process to generate this now. It's usually about 4 total steps to 5 steps. The first thing you do is you make this core. And this core is a semiconductor, and it's most commonly made out of cadmium selenide. That's called a 2,6 material. And the reason it's made out of that is, I'll talk about it later, it's mostly convenience because the chemistry works really, really easily. But also it gives you fluorescence over the visible spectrum, which is great. On top of that, you have zinc sulfide. And the purpose of that is basically to make it very, very fluorescent. Without this, it's not very fluorescent. It's a material with a wider band gap, meaning basically it insulates the whole thing. So it insulates the core, electronically insulates it. On top of that, you have some kind of coating. And that coating is what's kind of depicted here too. You have to have that for this to be used in biology because these have to interface with cells. And if you just have a crystal, that's going to just bind to everything. And so you have to have something that will make it stable and dispersed. And then you usually have 2 things on top of that as well. So you usually have polyethylene glycols. Everybody heard of PEG before? So PEG is used for a lot of things. It's used as part of consumer products, in cosmetics, and in lotions and things like this. But it's also used in biology and in medicine because it doesn't really bind to anything. And so you could use it as a non-stick surface. And so that's really important. Any kind of nanoparticle you ever want to put into a cell, it has to have a non-stick surface. PEG is the most common way to do that. It's basically a pretty inert molecule. And then you attach it to something, and that attachment makes it useful. If you just have this with only the PEG surface, you only have this particle that's dispersed in liquid and it's diffusing around. But if you can attach it to something that makes it biologically specific, then you can use it to study cells and study molecules inside of cells. Okay, so that's what the structure looks like. So now this is fluorescence. So this is the big property that makes them so useful. The big property is fluorescence. So this is a picture of 5 different vials of quantum dots containing liquid, which the quantum dots are dispersed in. And these are their emission spectra. So this means that these are different colors. Basically, the visible spectrum is somewhere in the range of 400 to 700 nanometers. This is purplish, bluish light, and over here is reddish light. And if you shine a light on them that's high energy, meaning short wavelength, you can excite these, and then they'll give off a certain color of light. So the big key property of this is, first of all, these quantum dots are fluorescent, but also their wavelengths are tunable. So you can see by these spectra, they're all distinct. You can get 5 different colors in the visible spectrum. What's great is that these are all made out of the same material. So these are all tuned by their size. So this is the diameter of these quantum dots, somewhere between 1.8 nanometers to 7.3 nanometers. You tune the color by the size. That's called quantum confinement. That's the big attribute that you get out of this. That's so useful. So if you've heard, yes?
So how exactly do you control the size?
That's a good question. So usually you synthesize these from a chemical synthesis. And the chemical synthesis, you basically start off with, if you saw the composition here, it's made out of cadmium and selenium. And so you start off with basically a chemical that has cadmium in it and a chemical that has selenium in it. You mix them together and they spontaneously start to grow a crystal of cadmium selenide, which is a gigantic crystal. So if you just mix them together, you'll get a gigantic bulk material out. But if you mix them together and you control it while it's growing, you basically form these tiny little seeds of just a few cadmium-selenium molecules. And then they keep growing, growing, growing. You can stop it at certain points. And then you get a certain size out. The way you do that is with these ligands. So these ligands bind to the surface as it's growing, and they prevent it from growing really fast. And so it slows it tremendously. And so you basically start off by mixing cadmium and selenium together. And they start off as a size that's like less than a nanometer. And then slowly over time, they start growing and growing and growing. And you basically stop it at one particular time point in which you want to have that specific size, which gives you that specific color. So it's basically a time-based synthesis. So that actually is a great point because, so you see, with just stopping a reaction at certain points in time, you can get different colors out. And so that's the only really type of fluorescent molecule that can do this. So there's two other types of fluorescent molecules out there that are really commonly used. One is an organic dye. And these are things like fluorescein, like cyanine dyes, rhodamine dyes. And if you work in a lab that uses microscopy, you probably use some of these. They're tiny. And there's also fluorescent proteins. Fluorescent proteins are very similar in their light-emitting structure as an organic dye, except that basically the organic dye is placed inside of a protein that can be expressed by a cell. And so you can get intrinsic expression of a fluorescent protein. So these two are fantastic for a lot of purposes, but it's a huge monumental challenge to tune the size, or sorry, to tune the color. To tune the color of fluorescent proteins from green, which was the original color that came out, to red took like 15 years. But to get quantum dots that emit from blue to the red, that's just a matter of size. You just let it grow longer. So that's a huge enabling capability. We can get really wide spectral ranges with a continuous tuning of color. You could pick any color that you want, basically. If you say, I want 405 nanometers, or I want 601 nanometers, you can pick that just by stopping the reaction at a certain point in time. Yes?
Are these Q dots toxic if you want to do like in vivo imaging?
You cannot inject them into humans. You can use them in mice for studying different things. But they contain cadmium. At least these do. So some of them do not contain toxic metals. Cadmium is a toxin. It's toxic to the liver and the kidneys. And so that's a huge field of study by itself, is understanding nanomaterials toxicity. It's thought that generally if a nanoparticle contains an element which is by itself toxic, then it'll probably never be used in a human being. Because if it gets in the body and it degrades, then you potentially can cause toxicity. So you can make these out of supposedly non-toxic materials. So you can make them out of any type of semiconductor. So carbon is a semiconductor. Diamond is a semiconductor. Silicon is a semiconductor. Those are all things that you can put in the body. There's a variety of them, and that's kind of a field in itself, is trying to create non-toxic quantum dots so you can use them for imaging. For our purposes, we never really inject these into animals unless we're trying to study some kind of effect. We're not trying to make them translational for use in human imaging. A lot of people are trying to do that. That's not our focus, but because I think it's a huge uphill battle to try to get these into humans, because it's like a 7-year process to get something from a new material that has proven no toxicity in mice to something that could actually potentially be used in humans, and then things fail through multiple stages of clinical trials. So if you're just using them, though, also you have to consider the fact that they're made out of toxic materials. That's a good point. So, yes?
So what's the cause of the wavelength change by changing its side? Is the energy structure changed?
Yes, exactly. So actually, why don't I go into that right now? Okay, so the reason this happens, that if you have a really tiny particle, it emits in the blue at higher energy, versus one that's really big, and emits in the red wavelength range. Higher energy or lower energy is because of the quantum confinement effect. So, does anybody know why fluorescence occurs in the first place? What is a molecule doing if it's fluorescent? What about if it's absorbing? What does an absorbing molecule do? Anybody know?
You're promoting electrons to higher orbital state.
Exactly, right. So every molecule has basically two things in it. It has got a bunch of nuclei and it's got a bunch of electrons. And for a molecule, you have bonding, and the bonding occurs through electrons. And those electrons can change in energy, and in molecules, they're called quantum energy states. So you only have specific energy states as defined by quantum mechanics. And so they shift in energy. So you can basically shine light onto a molecule, which has a whole bunch of electrons on it. And those electrons can absorb that energy from the light. And that's absorption. And then, if they're in a higher energy state, they can return back to their original ground state. And in the process, they've lost energy, and that energy gets converted into light. And so that's fluorescence. So that's what happens in a molecule. The same thing happens in a semiconductor. So the three defined states or types of solid state matter are generally considered to be, from the physical perspective, insulators, semiconductors, and conductors. What this depicts is electronic energy levels. So basically, in an insulator, what's shown here in blue is where you have electrons. These electrons that are at a lower energy level. And then you have another energy level above that at which they can exist. And in an insulator, there's this difference in energy between this filled electronic energy level and this empty one. So if you remember back from freshman chemistry, you have these HOMO-LUMO levels. It's the highest occupied molecular orbitals, lowest unoccupied orbitals. That's the same thing as this, but in a physical bulk solid material, you have these bands. It's called the valence band and the conduction band. That's the same thing as the highest unoccupied molecular orbital and lowest unoccupied electron orbital. So an insulator has a really wide band gap separating the valence band from the conduction band. And so what that means is that one of these electrons, if it's going to be excited, meaning that the molecule is going to absorb, it has to absorb a minimum amount of energy of this electronic band gap. Then an electron can be in this state, and then in fluorescence it can return back to its ground state by giving off light equal to this band gap. The same thing happens for a semiconductor, but for a conductor, you basically don't have a band gap. The conduction band and the valence band overlap. That's what makes them conductive at room temperature, is the fact that these electrons that are in this valence band can, just with very small amounts of thermal energy, move around the material easily because they're mobile in the conduction band. They're conductive. So semiconductors are some kind of intermediate between these two. That makes them really useful because this band gap is in wavelength ranges that's useful. So what happens then in fluorescence is, for a semiconductor, you have three-step process that occurs in fluorescence. You have excitation, where you have an electron somewhere in the valence band absorbing light, becoming higher in energy because of that, and entering the conduction band. And then in a really fast process, this electron returns back to its the lowest energy level of the conduction band. And then what happens is that it returns back to its ground state by returning back to the valence band, and at the same time giving off this wavelength of light that's equal to the band gap. So basically that means that whatever this band gap is, that's the color of fluorescence. So also note, this is important, that when you create this electron that's in this conduction band, it leaves behind this positively charged orbital here. So this electron transfers to the conduction band, and this orbital that was here in this band is now empty. And that basically is like a positively charged version of an electron. It's called the hole. It's an empty orbital. These are the two fundamental units of conduction when you think about conduction in solid state materials. Is when you create this electron in the hole, if you apply a voltage across them, these are what moves in different directions. So these are like the positively charged carriers, these are the negatively charged carriers. Okay, so that's why fluorescence occurs in a semiconductor. And this is why it's tunable. So basically you have this electron and hole. They're like a negatively charged particle and a positively charged particle. And as you might imagine, they'll electrostatically attract each other. And they do so in a semiconductor, which is a really big material. And they form what's called an exciton. An exciton is an electron-hole pair. And it looks almost exactly like a hydrogen atom. A hydrogen atom contains exactly one proton and exactly one electron. And that's actually exactly how we model this. We exactly model this from quantum mechanical perspectives as a hydrogen atom. So then what happens, this is for a bulk, large-scale semiconductor material. But then when you make this smaller and smaller and smaller and make this a nanoparticle, what happens are two things. First of all, this exciton has a specific size. It's in the range of about 10 nanometers for cadmium selenide. And so if you shrink this, what happens is that this electron in this hole, they have to be confined in space. They're spatially confined to a smaller region than they were originally. That's where the term confinement comes from. They're being confined in space. And as you get this particle smaller and smaller and smaller, it's confined to a smaller dimension because it only exists in the semiconductor itself. And so in the range of like a 1.5 nanometer quantum dot, you've shrunk the exciton from 10 nanometers to about 1.5 nanometers. So it's gotten really tiny. And so what happens when you do that is that the energy it takes to move an electron from the valence band to the conduction band increases. And so this is what's happening. This valence band is actually breaking apart. These now all of a sudden have specific electronic energy levels. And this band gap widens as the particle gets smaller. And that makes this tunable in the band gap by the size. And as you can see, you go from basically a red color for a bulk cadmium selenide to in the range of green to blue colors by shrinking the size of the nanocrystal. So that's where quantum confinement comes in, and it's also how you tune the color. Another interesting thing that happens is that you go from this really continuous electronic energy levels to ones that are really discrete. You see these energy levels coming out that are very, very specific. And these are like what happens in a molecule. And so these are the spectra you get of quantum dots as they're quantum confined. So this is a bulk cadmium selenide crystal, a really big one. And this is the absorption spectrum. And so as you move towards smaller and smaller and smaller sizes, you get these really discrete these discrete energy levels that show up. And the discrete energy levels indicate that it's more molecular. You don't really have these types of peaks in a solid material. They're usually only found in molecules. And so you shrink the size, you basically change the color, and you get more like a molecule. Yes?
Now, I've been noticing that you've been talking about the cadmium selenide layer because I assume that's the active layer in which the reaction is occurring. But you said earlier that you grow zinc selenide on top. So does that thickness affect the wavelength at all, or no?
It does, but to a much smaller degree. That's a good question. This core shell structure is actually, that was like the one example that's most commonly used. They're really broad examples of types of materials that are grown as core shells. And they give you tons and tons of unique properties when you do that. So the classical one of cadmium selenide zinc sulfide is this structure. Basically, that growth of zinc sulfide doesn't really change the color. And that's basically because it's a really just great insulator. It tunes it by just a couple nanometers, but it really has no major impact. You can grow other materials on here that have a huge impact, and that's a whole field called band gap engineering, and it makes these properties extremely tunable. You can move the locations of electrons to different areas within the core or the shell. You can move the electron in the hole independently, and that's really great because then you can make them into sensors. If the electrons are all at the surface, which you can tune that, it becomes almost like a plasmon band in a metal. And so they can become really useful for these certain types of applications. That's a good question. This shell is another semiconductor. And so if you imagine what's happening here is that we have a semiconductor core made out of cadmium selenide, and this is the energy level of its emitting state, this energy band difference. If we were to draw on here where zinc sulfide would be, these energy bands are like way up here and down here. That makes them an insulator, really wide band gap. And so we use that because if you put that wide band gap material on the surface, then the electron in the hole don't go into it. They stay in the core. That's called insulation. And so they basically are they're not impacted by anything that happens on the surface. And that's great because then they're brightly fluorescent all the time, which is a good thing for our applications. All right, any questions about that? So that's kind of how they work. So I'll take you basically briefly through the history of them. So nanoparticles made out of optical materials like metals and semiconductors have been around since the medieval times. Unbeknownst to the people that actually made them, the people that made pigmented glass for stained glass windows, a lot of what's actually in here are tiny colloids of metals and semiconductors. They're quantum dots and they're metal colloids. That's why they get these really vibrant properties and colors. They're stable over long, long times, centuries, because they are these inorganic crystals. So basically they were making glass, and if they put in small amounts of metals, small amounts of things like selenium, they found out they can get these really vibrant colors. This is actually the same way they made the first quantum dots in a lab too. So after that, there were a couple of reports in the 60s and 70s of various strange optical properties that were observed in labs. One of the first ones is by actually the Kodak company. So Kodak they call themselves, they went under like 5 years ago, but their company's kind of coming back under a different type of technology now. They call themselves the first nanotechnology company. That's because photographic film depends on silver colloids for development. And so they were in the field of nanotechnology back from the early 1900s. And so they had this paper where they published showing that depending on the size of silver halides, you can get different optical spectra. And so they didn't know what this was originally. They basically thought that something happened with the reaction. But they basically saw the size-dependent optical properties. At the same time, and also in the early 70s, people were trying to make semiconductors were part of a lot of early stage electronics back then. And then people started trying to go really, really thin slabs, really thin layers. And when they did so, they saw that these electronic energy levels changed when you make the thickness different. And so nothing really happened until 1981 and 1982, or sorry, 82 and 83, when two groups simultaneously reported the first ones that had really, really tunable properties. The first one actually was these two Russian guys that reported their findings in this really obscure Soviet journal that almost nobody read in the 1980s. And they defined quantum confinement, and they gave the fundamental physical principles for it, and they showed a great physical example in stained glass. So they basically grew nanoparticles in stained glass, got them different sizes, saw their colors changed. And there's a guy named Luis Bruce who was at Bell Labs then, At the same time, he was working on them, but he was making them in water. He made these cadmium sulfide crystals where he saw tunable electronic band gaps and tunable spectra by changing the size. And so in the 80s, this was kind of a pretty unknown phenomenon, and it didn't really catch on until the early 90s when there was major breakthroughs in synthesis. And so those synthesis breakthroughs really enabled you to make these tiny crystals over really wide size ranges to get tons and tons of colors out. The biggest paper was this one by Munji Boweni at MIT. He showed that you can make cadmium selenide with these really precise sizes over really broad ranges. That was a huge outcome. And so that kind of catalyzed the whole field, and cadmium selenide turned out to be the best one, and it still is the best one. That hasn't changed. It's kind of strange that still the original one from the 90s is the same one we use in 2016. So they weren't really useful though at that point because their fluorescence was really dim. They were usually in the range of like quantum yield. The quantum yield is basically when you have absorption occur and then fluorescence occur, it doesn't happen 100% of the time. The fluorescence might only occur like 1% of the time. And you have these other processes that compete, which you want to eliminate because those other processes are usually not useful. You basically generate a bunch of heat. And so that process efficiency is called quantum yield. And it was reported in '96 by this group at University of Chicago that you can increase the quantum yield from like 1% to over 50% by growing that insulating shell that I talked about, the zinc sulfide shell. That was the key step. That's what really makes them useful for biology. Without that, they're really not that useful. And then in 1998, simultaneously two science papers came out showing that these could be used for imaging cells. It was in the same issue. One of these was my graduate advisor, Shuming Ni, who was then at Indiana University. And then there was this guy, Paul Avasados from Berkeley, who's still a huge name in this field. And so since that time, 1998, there's been thousands and thousands of papers on the use of quantum dots in biology. So using them for fluorescence has been a huge thing. And so this field has really progressed in terms of what's been capable with them by changing the structure. So the first generation of these kind of looked like this. It was core shell structure, single ligands on them. The next generation that we're actually kind of still at right now, these quantum dots are sold commercially like this. They're pretty big molecules. They're attached to streptavidin. They're attached to antibodies for binding to things. And you can buy them commercially, use them off the shelf now. The third generation, we're trying to get around a whole bunch of the structural properties, and that's what we're doing in our lab. Ideally, we want them to be basically like GFP. GFP works extremely well to image things in biology because it's really small. It doesn't interfere with a lot of things. Sometimes it does, but very often it does not. We basically want to make them into this, but retain all their great properties. So, like you can see here in the previous slides, other things are fluorescent. It's not just the fluorescence that makes them so useful. It's the tunability in their fluorescence color. That's a huge thing. You can't do that in any other way without years of trial and error, basically, to make a new molecule that's fluorescent, like an organic dye, or to really, in high throughput, re-engineer fluorescent proteins. Those are really challenging fields where you don't have basic principles to work on. Just with basic principles, we can tune these colors way beyond the visible spectrum. You can go to the ultraviolet, infrared, to the mid-infrared. Yes?
So how do you determine for how long you can see this fluorescence, so when it dies off?
So that's one of the great things. So the photostability is tremendous. Generally, organic dyes and fluorescent proteins, when you're using them, use them in the dark, because even like room light, ambient light, can destroy your fluorescence. And that's irreversible. But these are basically infinitely photostable if they're made correctly. So we have some that are just sitting on the shelf for like 4 years and they're still fluorescent. And you can image them, like I'll show you in a second, at the individual molecule level for minutes to hours without a problem. That's one of the great things about them. So here's the great things compared to standard fluorescent molecules. Size tunable color, you could tune in the near infrared, which is great because you can't really do that with dyes very well. multiplexing. As you can see, these emission bands are really narrow. If you compare them directly with fluorescent proteins and fluorescent dyes, you see they're much more narrow and they're symmetric. They form like a Gaussian shape. That makes them really easy to deconvolve mathematically, obviously. If you have a spectrum like this compared to one like this, it's just a little bit more math to try to figure out how to deconvolve multiple colors. Since they're so narrow, you can get many colors in the same spectral range. So those are great, but in the last 10 years, the reason these have been so useful is because of these two properties: their brightness and their photostability. They're so bright, they're 10 to 100 times brighter than dyes and fluorescent proteins, and they're so photostable, you could easily see an individual one in a microscope. And so that enables us to see the most basic fundamental biology at the highest level of detail. So this is an image of a cell. And this is the membrane. The blue region is the nucleus. And these are individual quantum dots attached to individual molecules in the cell. So this is in a real-time video showing you what molecules are doing at the molecular level. So you could do this with fluorescent dyes and proteins, but you get an image in a video that lasts about 1 to 10 seconds. And then the fluorescent molecules are completely photobleached. They disappear. So quantum dots allow you to do this for in the order of hours. So you don't really you could just watch a process take place in real time for very long periods of time, so you can understand biological events at the most fundamental level of detail. Yes?
Do you see blinking in that, or is that?
You do, yeah, so that's a good point. So, so these individual molecules here, they look like they're not blinking if you just focus on one. And it's actually, in this case, we don't really have a lot of blinking because they're inside the cell. And so blinking is this phenomenon. Have you all talked about blinking in class? Okay, so blinking is if you look at an individual molecule, an individual fluorescent molecule, it's not continuously emitting light. And this is true for basically any type of fluorescent molecule. These are quantum dots that are just put on a cover slip of glass and then imaged with fluorescence. And if you just focus on any one of them, like that one or that one, you'll see it's flickering on and off repeatedly. That's called blinking. The process mechanism is still mysterious. So people have been studying this since it was discovered in the mid-90s. And so it's 20 years later, we still don't really know what causes it. We know how to control it now. You can control it by growing a really, really thick insulating shell. If you control that by doing that, it makes the particle really big, but it also makes it stop blinking. And so sometimes you want it to blink, sometimes you don't want it to blink. Sometimes it's useful, sometimes it's not. Generally in biology, it's not useful. It messes up your ability to track things. If you're watching a molecule move, you lose track of it when it turns off. So this is what you see on a cover slip, just on glass that's dried, or when you put fluid on it. When you put it in a cell, it seems to actually mostly stop blinking if it's inside the cell. If it's on the surface of the cell, it still blinks. But inside the cell, it's what's called a reducing environment. Inside of a cell, you have a lot of local density of electrons. And so this process is thought to be caused by ionization, where these quantum dots, they get excited by light, and their electrons go to a higher energy level. Some fraction of them actually shoot off and become ionized. And when you lose an electron, it basically turns off the quantum dot. And so it randomly returns back, the electron randomly returns back to it. And therefore we have this random blinking event take place. That's the current hypothesis about why this takes place. Inside of a cell, though, you have a high concentration of electrons because of the reducing environment. And so inside of a cell, it seems to not blink so much. But those are the great things you get out of this. So you can get single molecule imaging for long periods of time. So you could track these processes that we've never been able to see before. And so there's been a ton of biological discovery because of this in the last 10 years or so. Since these became commercially available, we could basically attach one of these to a protein and see what it does. So we could always infer what it did by, if you take the protein out of a cell and then you study it. You study its structure, see what it binds to, and you see what happens when it, is it an enzyme? Does it cause chemical reactions to occur? Now we can do it on a cell directly. And so we're using these just to take you through a few different things to image cancer. This is an image of a single cell, and we attached quantum dots to a growth factor that relates to cancer. So each quantum dot is attached to one molecule that basically tells a cell to move. And in cancer, cell motion is generally a bad thing. That's because that's the mechanism of metastasis, which is the spread of cancer around the body. And so we're trying to track these molecules at the single molecule level to understand what they're doing. So the idea is that if we could track them and understand them better, then we'll know how to treat them with drugs better. And so potentially you can stop metastasis from occurring. So this image basically shows you what these individual particles are doing, where they're moving. And then this looks like a bunch of random motion, but it's not. There's a whole bunch of order in here that's really difficult to just observe with your eye. And so we analyze all these movies containing these individual particles moving and see what they're doing. And so this is like the first step. You get out this image that shows you where is a heat map of where they're stationary. So when these particles become stationary, what's happening is that the cell's becoming activated in those regions. So if you track back these dynamically changing areas, like an area up here, there's a lot of stationary particles. One's over here, some stationary particles. Everything that's blue is really rapidly moving.
What's the time frame for that video?
This is real time. So this is like, I think, 10 frames a second. So this is what cells are doing. In a cell, this is how fast your molecules are moving on the cell plasma membrane. This is on the membrane. This is quantum dots attached to EGF, epidermal growth factor. And we're basically imaging this receptor moving laterally on the surface of the cell. This video that I showed before was on the inside of the cell. And this is also real time. So this is what molecules are doing inside the cytoplasm in real time. You see they're moving really fast. These are actually about 10 nanometer particles. If you shrink that down to a single molecule, like glucose, that's 10 times smaller. So these are actually, glucose is moving about 10 times faster than this. But things like the ribosome is like 35 nanometers. It's moving, it's really slow compared to this. And so basically those motions, we have to know what the size of the particle is in order to interpret what it's doing. So what we do after we get these videos is then interpret them by basic physical equations. This is the Stokes-Einstein equation. It relates something called the diffusion coefficient to the size of the particle. And we could basically watch a particle move and determine what it's doing based on this. We could determine whether it's bound to something. We could determine whether it is moving randomly or is confined in a small area. And that can tell us where it is in a cell. We could tell if it's in some kind of vesicle. We could tell if it's in some kind of, if it's in the cytoplasm or the nucleus based on this. And we do this by plotting this mean squared displacement. This mean squared displacement is a plot of the diffusion coefficient for each time increment. So basically if your particle moves a certain distance every frame, we plot this displacement. And we can see whether or not it's directed, it's confined, it's Brownian, basically completely random. And so that tells us a lot of information. And so there's been a whole bunch of biology discovered because of this. So one of the first things that was discovered in 2004 was by Diane Lidke at the University of New Mexico. Basically, this is the same biology that we were just looking at, the EGF and the EGF receptor. They found that you can observe these growth factors basically being intaken into the cell from long spindly fibers that extrude from the cell. So basically, usually thinking about a cell as kind of just being a fixed particle in place. It's got well-defined membranes. But a lot of cells that are, especially cancer cells, have long protrusive features. And it turns out that they use this kind of as like a probe to see what's around them. And they intake these growth factors from outward in, and they just basically pull them into the cell. These are the kinds of things you couldn't see before with molecular dyes, because you couldn't see them at the individual molecule level. And then another thing is neuronal biology. So neurons are one of the most differentiated cell types out there, meaning that they're most specialized and complex. A neuron looks like this. You have this cell body, and he's got this protrusive, elongated feature called the axon. And then you have all these dendrites that stick off from it, and they all have very specific purposes, and they're extremely complex. And one thing that would be great to know is exactly how all these independent groups work. So basically, this is just an information processing cell that transmits a signal from this end of the cell to the other end. And it does that by complex molecular machines. And so one of the other first things that was discovered was how neurotransmitter receptors get transported along the axon. And so basically, what you're looking at is molecules moving from this end of the cell along the axon. And so it wasn't known how they get transported. And it's really important because that dictates things like the learning process and the memory process and why that gets disrupted in Alzheimer's disease and Parkinson's. So imaging these has been extremely important for our understanding. Another thing is motor proteins. So motor proteins are really interesting because they consume energy and convert chemical energy into motion. And so we use these to track motor proteins like kinesin and dynein and see how they move at the individual molecule level inside of living cells. And so that couldn't be done for long periods of time with organic dyes. And so we can understand how motor proteins move along tracks, microtubules, inside of cells. One thing that just was released with our group and Paul Selvlin's group was understanding neuronal synapse biology. So the neuronal synapse is a connection between two neurons that's the fundamental unit of communication in the brain. So in the brain you have many, many billions of these that form the basic unit of learning, memory, and cognition. And in this you have basically one neuron's up here, one's down here. They connect through what's called a synapse. On the presynaptic cell, this is a cell that basically is giving information to the postsynaptic cell. And it does that by releasing these small molecules called neurotransmitters. They release them into this area between the cells called the cleft. And then on the receiving cell, the postsynaptic cell, you have these receptors that they pick up this neurotransmitter that was released. And what they do is they open up an ion channel and they cause an action potential, basically a voltage change in this cell. And so you go from having information here to having a voltage change in this cell, and then this cell can then communicate with another one. So the biology of this is extremely poorly understood in terms of the molecules structurally involved. The whole problem is that this whole thing is about 30 nanometers in size. So have you all talked about the diffraction limit yet of light? So what's basically the limit of resolution we can get with optics?
It's like 0.61 over lambda.
It's about half the wavelength.
Yeah, right. Yeah. So if you're working in the visible spectrum, for most of what we do is visible. If you're like at 600 nanometers, then 300 nanometers is the limit. So that's 10 times bigger than this. So basically, if you look at a synapse in a conventional light microscope, it just looks like a big blur. It looks like some object 10 times this size, but it actually has features that are much smaller than this. The synapse itself is 30 nanometers, but the things that make it up are less than 10 nanometers, so they're really tiny. And so quantum dots now enable imaging of this in a way that could not have been done before. The standard way of imaging these synapses is by electron microscopy. Electron microscopy, you get resolution in the angstroms, so 0.1 nanometer. You can see things at the single molecule level, no problem. But you have to kill the cell to do it. So you have to fix the cell, and then section it into thin sections, and you basically disrupt all the living biology in it. But now we can start to see these things with quantum dots. So we had to make these extremely tiny in order to be able to fit into this area between the cells, the synaptic cleft. So the big outcome of this was that in order to see these things, you have to have something like a quantum dot, which can emit light that you can detect at the single molecule level to see what it's doing when you're looking at these receptors. And it has to be extremely small. And so that's kind of a problem. Making these really small has been a really, really, really big challenge. This has taken about 15 years to do this. Quantum dots have been available commercially since 2002. And they've been really great to enable a lot of things. But they cause a lot of artifacts because they're pretty big. Their size is about 15 to 35 nanometers. So in that size range, that's much bigger than a protein. So if we're studying a protein with them, if your protein's this big and then your probe is this big, that's going to create this huge artifact of how it behaves and how it moves. And so because of that, we're trying to image these big quantum dots on these receptors. What you see is that this is an image showing where the synapse is. This is a blue region here, that's a synapse. And the red region here is where these receptors are, and they're moving. So this is like a long time frame movie showing where they are. They're basically always outside the synapse. And so people for the last 15 years have been thinking that most of these receptors are outside the synapse. But then when you look with small quantum dots, you see something completely different. You see they're almost all entirely within the synapse. This makes a lot more sense, right? These receptors are receiving neurotransmitters in the synapse. That's what they should be doing. And so people are trying to overinterpret this information that was actually an artifact for many years when it doesn't even make sense to begin with. And people knew that. They try to rearrange their whole framework of the understanding of the synapse based on this, when actually we should be observing something like this. So that's where we are right now with understanding single molecule processes in cells based on single molecule imaging with quantum dots. Okay, any questions? Yes?
So when you talk about the size, you're talking about the core of the shell and the ligands, not just, because if you change the core, then you just change the wavelength, right?
Exactly, yeah, it's everything together. What we're talking about with size is called the hydrodynamic size, basically how it behaves in water. And so if you absorb a ton of ions on the surface, you can make it bigger. If you don't absorb a bunch of ions, you make it smaller. The whole thing, it really derives from the surface. You can make the core really tiny. You can make the core like 2 nanometers. The surface coating has been the big, big challenge. So the surface coating is a whole bunch of complex events occur. Basically, there's 2 coatings. One is called a polymer coating. And it's basically coats it in a micelle, and a micelle is basically encapsulates it inside of a big polymeric shell. This is what's sold commercially, and they're great because they're ultra stable off the shelf for years. You can make them really tiny like this with just a really thin coating, but they decompose extremely rapidly. And so being able to make something really tiny and stable has been a big problem. And so we did this by making these coatings that are called multidentate polymers. It's kind of bridges those 2 extremes of a really big polymer coating and then a really thin single molecule, where we basically took these polymers and then fused them to the underlying layer. And you get this really thin coating then. And so what happens is that you basically go from something like this, that's like in the range of 10 to 15 nanometers, to one that's like this, which is in the range of 5 to 6 nanometers. So 5 to 6 nanometers is kind of ideal, we think. That's approaching the size of green fluorescent protein, and it's basically the size of most proteins. So this is a continual effort. People are trying to make these smaller and smaller and smaller. Not just for imaging like this, but for almost any application you want them to be smaller, especially if you want to inject them in the body. So you asked before about toxicity. Theoretically, you can inject anything that's toxic into the body as long as it fully eliminates. And so we already do this. If you get an MRI, the most common contrast agent they inject you with is a gadolinium. Gadolinium is extremely toxic. However, it's bound to a molecule called a chelator, And when they inject it into you, something like 99.99% of it goes out into the urine. And so you don't have to worry about it. And so you can inject really, really toxic things in the body as long as you can completely eliminate them. And that's really well known that it works really well. But it's also known that nanoparticles, as long as they're small enough, they can get out of the body. And so making them really small is really important. So this shows, this is the bladder, and up here are the kidneys. And this fluorescence that you see here is quantum dots in a mouse. Basically what happens is that if you inject a quantum dot that's small enough into the body, into the bloodstream, it will get out of the kidneys and into the urine, and it'll leave your body entirely. But there's a specific size threshold for this. It's about 5 nanometers. And so you see 4.4 nanometer particles efficiently removed from the body. The bladder fills up with fluorescence. 5.5 to 6 is a really strict cutoff. It turns out making something this small is an extremely difficult engineering challenge. Because if you imagine all the components that go into it, you've got a core, you've got a shell, you've got this coating, you might have something attached to it to bind to something. Getting something that's that compact and precise is not easy. And so people have done this in proof of concept by creating these artificial systems that do this really well. But this has not become practical yet. So this is a long-term goal. It's making them smaller than 5 nanometers. We made them about 6 nanometers. That 1 nanometer change is immense. So it's really difficult to get them that small. Okay, that's probably about everything. Does anybody have any questions? This is a 1-hour class, right?
Goes until 12:20.
12:20? Okay. Sure. Well, nobody. Any questions at this point? Okay, so I could talk about.
I have a question.
Sure.
So basically you can tag anything in regards to cancer?
Sort of. So you can tag things that are accessible. So you could tag things where there's no physical object in the way. So that means things outside the cell. So anything that's a macromolecule, like in the range of bigger than a nanometer, doesn't spontaneously enter a cell. And so you could tag anything outside the cell that you want to. And so that means receptors, and that means things like lipids, and anything that's accessible like matrix proteins. But things inside the cell, that's a huge challenge to get things inside of a cell. The whole field of drug delivery has been limited by this for like a century. Getting a macromolecule inside the cell is not easy. So if you want to evaluate something in cell culture, it's easy to get things in cells. There's a whole slew of methods to do this, like microinjection. We use one called osmotic delivery, and you can get them in the cells in culture.
But if you have a piece of 3D tissue, muscle, whatever, how do you administer that to that scenario?
You can't, as far as I know. So there's ways to do it. So I don't know if I have slides on this. Let me see. So there's a whole, no, I don't have slides on this, but there's a whole bunch of methods to deliver things to cells. You have physical methods like, we could just write these up by like method, by the physics of them. You have physical methods. So microinjection. Actually, it's probably better right over there. So microinjection. Where you have a cell that's sitting on a surface, and you literally take a micropipette and inject fluid into it. So that's how embryonic nuclear transfer is done, and that's how basically whenever you want to put something inside of a cell really efficiently and know it's in the cytoplasm or the nucleus, you could do that. That's the standard way. As you can imagine, it's extremely low throughput. You do one cell at a time, really high failure rate. This pore clogs all the time, a whole bunch of problems. And then you have electroporation. And you basically use an applied voltage to basically disrupt this membrane, and it breaks apart. If you control it really precisely, it doesn't kill the cell. But that means that anything outside the cell can get into it when you have electric pulse applied. Then you have chemical methods. So I'm distinguishing these because these you can clearly do on like cell culture on a flat dish with cells with liquid on top of them. You can't do them in like a 3D tissue. Like even if you have like a 3D tissue that's taken out of the body or you create one, you can't really do these efficiently. But you can do this with chemical approaches. And so what you do is you package them inside of a material that then puts them into a cell. And so there's a whole bunch of methods to do this that have been developed for gene delivery that you can take and apply the same methods for quantum dot delivery. And so you can encapsulate them into something that's like a liposome, which is a whole bunch of lipids. And it basically serves as like a delivery vehicle for getting into a cell. And it causes membrane fusion. So if you put this on a cell, oops, and it fuses with the membrane. So basically this binds to the surface, and you have a quantum dot inside, and then it releases it into the cell. Basically, this membrane fuses and you get inside here. This is extremely inefficient. So this is the reason why gene and drug delivery is still a problematic field, is because of the efficiency of this process. There's other ones that are called peptide approaches. So if you take a quantum dot, you normally coat it with PEG, like I said before. That's the molecule that makes it inert and makes it not bind to anything so that it can perform a unique task. You can attach it to a cation, so a whole bunch of positive charges. It turns out almost everything in biology is negatively charged. And most proteins are negatively charged. The plasma membrane of cells is negatively charged as a net negative charge. So if you attach it to something that's positively charged, it makes it bind really, really specifically to cell membranes. If you do it in a really controllable way, you can get it to interface with the cell and then rapidly enter. And so that's more of a, it doesn't have membrane fusion occur. You deliver through an endosome, so basically the cell engulfs it. And then this whole thing can break that whole thing apart. It can break the endosome apart. So there's another way, so we've been working with another way to deliver things to cells that by tuning the size and shape. So these, we just reported these, was if you have a quantum dot that's like a sphere, it doesn't really interact with a lot of things. But then if you make it different shapes, you can tune that interaction. So we wanted to make a way to really efficiently get quantum dots inside of cells. So we changed the shape into a flat surface. So that quantum dot that we showed before, that's like a sphere crystal, we basically flattened it out. And if you're familiar with anything that's used in optical electronic devices, this is now called a quantum well. It's a really flat sheet of semiconductor. In the dispersed form, they're called nanoplatelets. And so they're basically a really thin sheet of cadmium selenide again. And they've got ligands on them like they did when they were spheres. You can grow on another insulating shell, just like you normally do, to make them fluorescent. And then we encapsulated them inside of a lipoprotein. So lipids, and there's something called a membrane scaffold protein. This is very similar to that polymer encapsulation strategy. And the key outcome, well, they're still fluorescent and everything in water. So once they're transferred to water, they're fluorescent. But that requires that they have this coating on them, that they require this insulating shell. Oops, I didn't put in the slides that has the cells. Oh, so anyway, they really efficiently enter cells. That's the big outcome. Well, Well, if you're interested, I can send this to you. This is published in Journal of the American Chemical Society. Basically what happens is that these enter cells extremely rapidly, but they're also, if you do it too fast and too much, they're extremely toxic. So what happens is that this flat surface binds to the flat surface of the cell in a way that you can't get with spheres. You basically get a lot of surface contact, and that causes the cell surface to basically change really rapidly, and it takes up the entire particle extremely rapidly. This doesn't happen with a sphere. A sphere, you have really high surface curvature, and if you're trying to think about a flat surface of the cell and a sphere interacting with it, there's only like one point of contact. So you've got a really weak total interaction. But when you flatten the surface out, you get tons of interaction. And so that's like a surface adsorption effect. So there's a lot of ways to deliver things to cells. None of them have been totally effective yet for getting things rapidly into the cytoplasm. So that's one place where fluorescent dyes and fluorescent proteins have a major advantage. Fluorescent proteins can be expressed in a cell. So they're actually, the proteins are being produced by the cell itself and put anywhere they're supposed to be. All right, any other questions? Yes?
Are these just as bright as the sphere ones?
Good question. They are. So we tested that. So it all depends, the brightness depends on two things. The quantum yield, which is basically the conversion rate from absorption to fluorescence, that can be in the same range. It can be like 10 to 90% depending on how you make it. The other component of brightness is how much it absorbs at any one particular point in time. That actually is what dictates this whole thing. So it's really all about absorption efficiency. And we found that this is the brightness at the single molecule level for these. And if you compare a nanoplatelet versus a quantum dot, they're almost identical at the same wavelength of emission. Of course, these are very tunable properties. The brightness is basically dependent just on really the size. So that brings up a really good point. So brightness is an extremely important parameter for these. You think about the fluorescence wavelength. You could tune the color. You can make different wavelength ranges for detection. That's great. But if it's really dim when you do that, it's not that useful. And so that's actually been a huge problem. So we had a paper on this in the fall that describes how to engineer all this. And so this is one thing that really excites me in general about quantum dots, is that if you want to change a parameter, there are basic principles already written in physics textbooks and in the semiconductor industry exactly how to do it. We know exactly where all these parameters derive from. You can't really do that with a lot of other optical materials. It's really difficult to predict. You can predict things like absorption really well, but fluorescence is really difficult to predict, except for quantum dots. So this is the concept of quantum confinement. Blue, green, red, you get these different colors by tuning the size. And that's great. You can tune the colors many different wavelengths all across the visible spectrum. But a big negative attribute of this is that because the sizes are different, you get different brightnesses. These really small ones simply absorb less light just because they're smaller. The really big ones absorb a lot more light more efficiently. And because of that, the difference between a green quantum dot and a red quantum dot in terms of absorption efficiency, which translates to brightness, is a 2 orders of magnitude difference. So your red quantum dots, which are shown here at the single molecule level, are 100 times brighter than blue ones. These can barely be seen. And when you do like multicolor experiments, this is what your result is. If you mix them together, these red ones significantly outweigh these green ones when they're supposed to be detecting at equivalent levels. So that's a big problem, not just because you have a difference in signal-to-noise ratio. This is a huge problem because in this spectral range, you have autofluorescence. So cells are fluorescent naturally. They're fluorescent in the green spectrum, and so they completely overpower these green quantum dots. So as a result, almost everything you see published uses quantum dots in this spectral range, 600 nanometers to 650. They almost never use these 300 or these 500 nanometer particles. They're just simply not that effective. So there are fundamental ways to get around that by re-engineering the particle material. So that's why we developed these new materials that are called brightness equalized quantum dots. So you have size-tuned quantum dots, which is based on the quantum confinement principle. And the big thing is that you have different sizes yields different colors. But at the same time, this parameter is your extinction coefficient, which is basically the same thing as absorption. Absorption efficiency is hugely different across different colors. So we change this by a new structure, which has 3 different materials in it. So it has a core and then 2 shells. And what this allows you to do is that we can tune the color with the same size as long as we change the composition. And so you could do that by alloying. So we created these alloys where we had cadmium selenide originally. Then we alloyed it with mercury and sulfur. And that gives you this continuous tunability of the band gap, independent of size. So now we have an independent way to tune the wavelength apart from size and brightness. And we actually included this second layer to independently control the absorption. And then this third layer, as always, controls the quantum yield. So now we have independent parametric control of these 3 different things, which, as before, they were all coupled together. And so you couldn't tune all these things before. So this gives you brightness equalization. And so this translates from the level of the fluid level. This is the brightness values. Basically, this peak here indicates the brightness of a red quantum dot versus a green one for the size-tuned particles. And as you can see at the single molecule level, the red ones the bigger ones are very bright compared to these green ones. It's hard to see these images here for the brightness particles that are equalized. They have similar brightnesses at the ensemble fluid level and at the single molecule level too. So you can see that just by visually looking at them. If you shine a laser at brightness equalized dots versus ones that are the size-tuned mismatched brightness ones, the red ones overwhelm in the size-tuned ones, but you can see easily the greenish bluish ones otherwise. So that's neat and all. We get better use of the green spectrum. But it also, for our applications, we want to understand biology better. And this gives us a new tool for quantitative imaging. So the big outcome of this was that we injected 2 colors of quantum dots into a mouse bloodstream. And we tried to figure out if we could quantify them. Can we count them? It turns out if you try to do this with size-tuned quantum dots, your expected value, this gray value here, is very different than what you observe by about a factor of 3. So even if you know all the properties of their emission, their mismatched brightnesses give different results than what you'd expect and what actually is there. So this is because of all the other optical things that happen in tissue. You have scattering that occurs, and you have absorption that occurs, and you can't really control that. But when they're brightness equalized, you get almost the same value out that you expect. So basically, we can now do quantitative imaging in thick tissue of molecules. We can know for sure that we have a certain number of molecules. When we detect them, we know exactly how many are there based on their matched brightness values. So that was just published last year. You can see that paper here if you want to. Anybody have any other questions?